we were studying factors affecting enzyme action, right? So there are 6 factors we will consider in this class. First thing already I have explained in the last class, enzyme concentration. So that says the rate of reaction will be directly proportional to the concentration of free enzyme, and it will go on so unless substrate is a limiting factor. In case substrate is a limiting factor, it will maintain a parallel line after some time, when all the free enzyme, all the substrates will be engaged by the free enzyme. Right. Next comes temperature. As we know, our body temperature is maintained at 37 degrees Celsius roughly. So all enzymes must function optimally, not maximally, optimally at 37 degrees Celsius. Correct? Otherwise, we won't be able to digest or metabolize any of the food. So most of the enzymes which found in our body within the cells, remember, not outside cell, most of the enzymes, 99.99 or 100% enzymes within the cell and 99% of enzymes outside the cell have their maximum activity near between 37 to 40 degree, between 37 to 40 degree Celsius, correct? So with increase in temperature from 0 degree onwards, you say, the enzyme activity slowly increases. How do you describe? First you draw the, how do you write the answer? First you draw the graph, then explain the graph. That's all, nothing else. Correct? So how do you explain this graph? The rate of reaction increases gradually with the rise in temperature. First sentence. Second sentence. It reaches maximum at 37 degrees Celsius. Stop. Third sentence. The rate of reaction rapidly decreases. I ki bolechhilam gradually. Ekhane ki bollam? Rapidly decreases with further rise in temperature, or rather beyond 40 degrees Celsius. 40 degree Celsius ke যদি আমি ফারেনহাইটে কনভার্ট করি কত হয়? Convert 40 degree Celsius to Fahrenheit. একটা চড়াবো, ঠিক করে কর, জানিস ফর্মুলাটা মনে আছে? বল কি? C/5 is equal to F minus 32/9. কর। Yes, 104. So what is the maximum temperature shown in the thermometer, clinical thermometer? 104 the maximum, correct? এর উপরে জ্বর আমরা মাপি না, কেন কি? 40 degree is the maximum beyond which person must not survive if they persist for a long period, তাই না? So দেখো, তাহলে জ্বর বা ফিভার মানে কি হাইপোথার্মিয়া, তাহলে হাইপোথার্মিয়াতে আমাদের এনজাইমস ওন্ট ওয়ার্ক। যখন আমাদের জ্বর হবে বা হাইপোথার্মিয়া হবে, তো এনজাইমস অ্যাক্টিভিটি উইল র্যাপিডলি ডিক্রিজ। সি within 10 to 15 degree it decreased to 0, and whereas it took 40 degrees Celsius to reach maximum while increasing. So now 3 sentences done to explain the graph. Now what is the mechanism behind? We know in a solution when we increase the temperature, what increases? The effective kinetic energy of all the participating molecules in the reaction. Correct? কি বলে ব্রাউনিয়ান মোশন, তাই তো? সো উইথ ব্রাউনিয়ান মোশনের থিওরি কি ছিল? টেম্পারেচার বাড়লে ব্রাউনিয়ান মোশন বাড়বে, হাফ mv স্কয়ার, correct? তাহলে when we increase temperature, the enzyme and the substrate both will increase in their speed. Why speed is important? Because with more speed they will collide, more amount of energy will be dissipated from kinetic form to static form. The static form will change the shape and size of the substrate and bring about the changes to convert it to product. Correct? When they collide, if they collide slowly, then the exact amount of energy will not be dissipated. When they collide with optimum speed, that energy will be, you know, suppose I keep this. Did you hear any sound? No? Why? What is the difference? Half mv square. I just increase the velocity. See, sound increased. Right? Similarly, the same energy is converted to static form. Here, kinetic to sound, and in this reaction, kinetic to static, strain. Right? So, with rise in temperature due to rise in the effective velocity of the participating molecule, the rate of reaction will increase. It will reach maximum. Then why not? It will go on increasing even after 40 degrees Celsius. If it increases to 50, 60, 70, 80 degrees, then more speed, more reaction should have happened. Why? Because at higher I will lose my job because of it. Because at higher temperature, the protein will lose its structure. It will get denatured. So what will happen? It will never have. It will no longer have the desired function, because we know that function of a protein is a function of its structure. Correct. Correct. Same thing. So always try to practice this question. It is very, very important. Very often this is asked. So, are there any enzymes which are exceptions of these rules? Yes, there are certain enzymes which are found in the hot spring bacteria. Correct? They can survive even beyond the temperature 75 degrees Celsius. Right? And that's why they are used in PCR reaction. Next comes pH. Now in the pH, you have to remember at least 3 examples. One showing optimum pH in the acidic range, one showing optimum pH at the physiological pH range, and one showing optimum pH range at alkaline or basic range. Correct? Now what does pH affect the rate of reaction or enzyme activity? We know that the substrate binding site, what is that substrate binding site? Is a portion of active binding site which actually holds the substrate in position prior to reaction. Correct? So the substrate binding site contains what? Ionic residues or hydrophilic residues. The pH of the medium alters the ionic state of these participating amino acid residues present in the substrate binding site. I repeat, the pH of the medium alters the ionic state of the participating amino acid residues present at catalytic site. Catalytic site. Correct? So because this happens, so previously which was positively charged, now due to high pH it has become negatively charged. So what will happen? It will no longer hold the substrate in position, so rate of reaction will decrease. Correct? So it depends on the nature and pI of the amino acid residues present at the catalytic site. Now you understand what is the importance of pI. If it is fall under pI, then what will happen? It will have no longer any charge, so it will not function. So the catalytic residues will not participate in catalytic process. So here we keep some examples. Some pepsin have their optimum pH between 1 and 2. Between 1 and 2. Remember, these two questions are different. What is the pH of gastric juice and what is the optimum pH of pepsin? Correct? These two you must not mingle. Answers are not same. Correct? So the pepsin function maximally between 1 and 2. See, 1 and 2. Salivary amylase around 6. Around 6. Correct? Alkaline phosphatase. What do you find? Now all these enzymes are what? Digestive enzymes. That means they are ectoenzymes. They are ectoenzymes. Ectoenzyme means which are secreted outside the cell in GI tract. Correct? So these are ectoenzymes. So they have varied optimum pH. Alkaline phosphatase has pH around between 10 and 12. Between 10 and 12. Correct? Now we come to endoenzymes, means which are present inside the cell. Inside the cell mostly the pH is 7.4. So what will happen? All will have similar optimum pH, which is equivalent to physiological pH. Correct? But still few enzymes will differ. So for example, acid phosphatase. Acid phosphatase. What is the optimum pH? It is between 4 and 5. So just add acid phosphatase here. Between 4 and 5. Having optimum pH between 4 and 5. Never mention exact value. Give the range. Okay. Done? দেখা গেছে সবার? Now, next is effect of substrate. Effect of substrate. Now, these here it comes the biggest problem, because in this particular factor we have to calculate enzymatic reaction rate, amount of product formed. So all sorts of problem came in this factor. Right? What is that? They found the rate of reaction increases with rise in substrate. Remember, in this case, enzyme is a limiting factor. Means enzyme concentration is fixed. Correct? So they will start increasing the substrate. You will see slowly, gradual increase in rate of reaction. Then it will increase to maximum. Slow, much slower. Then it will become parallel. However, more substrate you add to the system, the rate of reaction will remain same. Rate of reaction will remain same. That doesn't mean reaction will stop. Reaction will continue, but the rate of conversion of substrate to product will remain same. Right? So this kind of graph depends on molecularity of the reaction. You have any idea about molecularity of reaction? Okay. Now cite one example, enzymatic reaction, and tell me what is the molecularity over there. Enzymatic reaction. এই যেমন পেপটাইড প্লাস পেপসিন, তাই না? এরকম টাইপের। বা ল্যাকটোজ প্লাস ল্যাকটেজ, গ্লুকোজ প্লাস গ্যালাক্টোজ। মলিকুলারিটি কত? কে বলতে পারবে? কত অর্ডার? Which order reaction is this? Order of reaction. So, which order reaction is this? Lactose to glucose plus galactose. Which order reaction is this? Ball, the ball. So, Jama. It is lactose broken down into glucose plus galactose. What is the order of the reaction? Huh? Next. Phosphorus reaction. Kind of phosphorus reaction? Hey, order of reaction is formula key. Product. Key. Formula key. Is enzyme, now my question is enzyme here is a participant in the reaction or not? Huh? Yes, it is. This is a difference between chemical reaction and correct. So molecularity or the order of reaction changes. So here, sit down. So higher the order of the reaction is, this graph will be more slanted towards x-axis. Lesser the order of reaction will be more perpendicular to the y-axis. So in case of zero order reaction, it will be hyperbolic. In case of two orders, first order reaction, second order reaction, third, fourth, it will be further slanted towards x-axis. This is a basic thing you must understand. So when they started this particular theory to explain, they came up with this problem, whether to consider enzyme as a participant or a spectator. In the class, I am participating, few of you are participating, and rest are spectator. They are either participating, not listening anything, they are just seeing what others are doing. So what is the role of enzyme here? So enzyme is always a participant. And that was a mistake first done by the initial chemists who had idea of organic chemistry reaction or inorganic chemistry reaction, where enzymes do not participate in the reaction. In the first definition in the enzyme or catalyst was what? Catalyst do not participate in the reaction, it remain unchanged, at least chemically, if not physically. After the reaction, it either initiate or stop the reaction. All things are fine, but with enzymes, they participate in the reaction, and after reaction, it remains unchanged. The difference. So they came up with a formula which is called Michaelis and Menten equation. Michaelis and Menten. They first coined this term. No, they didn't coin the term. They discovered the formula. Now, after 50 years of the discovery, Briggs and Holden brought about some changes in the formula and then named it by the discoverer. That means like Dalton's theory. Most of the theory is wrong, but still we study. Why? Because of the historical importance. Right? So they, Briggs and Holden, JBS Holden and Briggs, they brought about some changes in the formula and then named the same formula after bringing the changes as Michaelis-Menten formula or equation. Right? This is the history behind. Now, the rate of what you have to write here, you have to draw the graph and then correct? This you have to write. I will tell you what you have to read and what you have to not. Maximum activity occurs when the enzyme is saturated. That means all enzymes are occupied by the substrate. Where? At this level. All enzymes, these are enzymes, all enzymes are occupied by the substrate. So no more free hands. Exponential and asymptotes levels of. মানে slowly বাড়বে, বেঁকে যাবে, it is not straight line. এবং asymptotes মানে বেঁকে গিয়ে প্যারালাল হয়ে যাবে। হলো? Now write this. This is Michaelis-Menten equation. V0 is equal to Vmax within third bracket S. Within third bracket মানে substrate concentration. Km plus, এবার এটাকে মুখস্থ করে ফেলো। By heart it, right now, right here. ঠিক আছে? V0 মানে initial velocity. Vmax মানে maximum velocity, মানে when all the enzymes are, তাহলে আমরা একটু গ্রাফটা ফিরে যাই। এখানে দেখো Vmax কোথায় বলো? V0 কি? ইনিশিয়াল ভ্যালুসিটি 0-তে, বাড়তে 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 এখানে যাবে ম্যাক্স। কেন? এখানে আর বাড়ছে না, এখান থেকে একটা স্ট্রেট লাইন টানবো, হ্যাঁ, এক্সট্রাপোলেট করলেই ওখানে আমরা Vmax পাবো। হলো? এবার এখানে কি দেওয়া আছে? Km. What is Km? Definition? পড়ো, পড়। তোরা কি বাড়ছে নাকি? আমি ওখানে দেখে দেখে পড়বো, তুই শুনবি তাহলে, রাইট? অ্যাকচুয়ালি মজার ব্যাপার হচ্ছে Michaelis and Menten did not name it as Km. They named it as Ks. It was Briggs and JBS Holden who named it as Km. That means some changes were there in the constant. So they changed Ks to Km. Something they changed. What did they change? I tell you in a very short and easy manner. You need not to know all the detail, but just know what was the difference they added. Now, this is a reaction, okay? What Michaelis and Menten said in this reaction, এখানে কি আছে বলো? কতগুলো কনস্ট্যান্ট আছে? কি আছে? K1, K minus 1, K2, okay? Take a reaction where enzyme combines with substrate to form enzyme substrate complex, then it has equal possibility to go in either direction. Why? Because this is a transition state. So E plus P can be formed, or E plus S can also be formed. If it forms E and P, then this will have constant like this, and the moment it will form E and P, by the law of reversibility of all reactions in this universe, all reactions are spontaneous, all reactions are reversible. Right? Suppose if you keep lactose for 100 years, it will be cleaved automatically to glucose plus galactose. That is a theory. But can a child wait 100 years to grow? No. Can you? Right? So we must accelerate the reaction, and then we must maintain the direction of reaction. A child is breaking the lactose to glucose plus galactose, and immediately it is formed back. Then what will happen? The child will always keep crying. His hunger will never be satisfied. So immediately that glucose should be utilized by the further reactions. Then only reaction will go on in particular direction. Right? So Michaelis-Menten said বলছি এই কনস্ট্যান্টটা অনেক বেশি, মানে রিয়াকশন হয়ই না। Practically, there is no reaction at all to form product. This was the first concept they gave. So, if it is very low, then there is no question of this. If the product is hardly formed, there is no question that product is formed back to enzyme substrate complex. So they said that K2 correct. Now what is the constant here? What is the constant? K1 plus K2 by K2, K1 minus 1 by K2. Oh, sorry, K2 by K1 plus K minus 1. Okay. Then Briggs and Holden, they said K2 in biological system it is not correct. Right? They said a lot of product is formed and it is very rapid reaction. So K2 is cannot be ignored. K2 cannot be ignored. So they form like this. Sorry, it is minus 1 by K1. This was by Ks. Km became এটাকে দিয়েছিল Michaelis-Menten. K2 নেই, তাহলে যা হচ্ছে এইটার এইটা K minus 1 divided by K1, যা তৈরি হচ্ছে সেখান থেকে আবার সবটাই প্রায় ব্যাক করে আসছে। ও বলো না K2 আছে, তাহলে ES এর now ES will have two fate. It can go to either direction, this direction as well as this direction. So utilization of ES will be cumulative factor of these two. Correct? And then formation of ES will be K1. So they named it as Km. And they kept the name as Michaelis constant in view to show respect to their predecessors. That's all. This is the difference between Km and Ks. And this is called Briggs and Holden modification. They first introduced steady state theory. What was that? See, this is the graphical determination of Km. Km means half the substrate concentration at half the maximum velocity. Substrate concentration at half the maximum velocity. Substrate concentration at half the maximum velocity. মনে রাখবে, ভোলার কোনো চান্স নেই। Substrate concentration at half the maximum velocity. Why Km is important? Because Km, by knowing the Km, we can understand how much affinity of an enzyme is having towards its substrate. Now this particular graph shows the steady state kinetics. What is steady state kinetics? See, in this graph there are 4 lines. One line represents the substrate concentration, another line represents the free enzyme concentration, Another line represents the product concentration. Another line represents the ES or enzyme substrate complex concentration. 4 lines represents 4 types of concentration. Okay. At the beginning of the reaction, when all of the enzymes are in free form and all of the substrate are in free form, then substrate concentration is highest, red color. Enzyme concentration is highest. What is the color? Light purple. Correct? Huh? Then after some time, the substrate concentration will start fall. Why? Because it is being utilized. At the same time, the product concentration will start increasing, the blue line. Then, at the same time, enzyme concentration, the free enzyme concentration, is started to decrease, but not zero. Why? It cannot be zero. At least some or more, and some or other enzyme will remain free, otherwise reaction will stop. You have to freeze the reaction. But we are not freezing the reaction. We are allowing the reaction to grow, but we are measuring the rate of reaction at every moment, a given interval, say around 30 seconds, 40 seconds, 50 seconds, 1 minute or so, like that, depending on the speed of the reaction. Right? So now we find this graph. The ES complex and E maintains same rate, between this point and this point. What are these points? The points are the point where the substrate is started being utilized, and substrate is completely utilized. These 2 points. Right? So in between these 2 points, if we measure the rate of reaction, it will show no change. That means it's maintaining a steady value. That's why we call it a steady state. But is it like reaction has stopped? No, reaction is continuing, but rate has come to a standstill. But reaction is continuing. That's why this is called steady state kinetics. Is it okay? Good enough to understand? See, you must understand this concept. Then it will be easy. You need not to mug up. You need not to derive the Michaelis-Menten equation. That is taken away from the syllabus. Even if it comes in the examination, that will be out of syllabus. If you can recall, I told you in the first day, read your syllabus very carefully. Read your syllabus very carefully. It is changed from last year. So read your syllabus very carefully. Okay. Free enzymes lowest, 0 to hobe na. He represents an enzyme. Now he is telling, why not enzyme becomes 0? So that all the enzymes are participating in the reaction. Then only it can reach maximum. Now what I will do, I will throw this mobile and he has to catch without dropping any one of those. Ready? My mobile. Right? So what is the concept? If the hand is not free, how the reaction will proceed? So it will maintain a basal concentration, base amount, right? Which will be free. Enzymes must have their hands free to hold the next substrate. Otherwise, reaction will stop. That's why some amount of enzyme will remain always free. That's called basal amount. The actual concept of basal amount is unexplained. When we cannot say what is it, we say basal amount. Huh? Now, see, inside the cell, there is high potassium and low sodium. Correct? The question arises, who let the first potassium inside? It should be, theoretically, it should be 0 potassium. Huh? If you add more sodium outside, potassium will be 0. But that never happens. We don't know. We say basal amount. It is still to be discovered. See, I told you, I gave you a chance to win a Nobel Prize. So these are the assumptions taken by Michaelis-Menten. They thought reactions go like this, but they were not correct. Later on, it was proved. You know, there is this JBS Holden. He was a very renowned, very big scientist, biochemist by nature. But he didn't receive Nobel Prize, but he was a person who could have received Nobel Prize. Anyway, there's a road in our city, Kolkata, by his name, JBS Holden Avenue. Actually, he was in US, but then he was moved by Marxism, and then he came to Kolkata because that time Marxist government was there. So he worked there, he liked the city, he stayed here, and then he went off from here, but then after his death, we kept a road by his name. So, because he chose to show interest to the Marxism, America never gave him Nobel Prize. Probably. I don't know. How would I know? I was a very small kid that time. Very small. Literally very small. Done? Okay? Don't write. This is a derivation of Michaelis-Menten equation. It is omitted from your syllabus. So enjoy. Now you remember this. What? You remember that form? V0 is equal to Vmax into S divided by Km plus S. Now you remember this form. 1 by V0 is equal to Km by Vmax into S plus 1 by Vmax. Correct? So this is called double reciprocal plot or Lineweaver-Burk plot. Now there are 3-4 such plots. One of them is Lineweaver-Burk plot. LWB, Lineweaver-Burk plot. Lineweaver-Burk plot, like this. What is the problem with this previous equation? V0 is equal to Vmax into S divided by Km plus S. The graph was exponential. Now, which graph is easier to calculate? A straight line or an exponential? Straight line. So, we changed the nature of the graph to straight line. That's what? So that we can easily calculate. This is the need of discovery of this. Are there other methods? Yes. There is another method called Adey-Hofstee plot. Right? Similar thing, but in a different way they plot the x-axis. Right. So now this is y is equal to mx plus c form of straight line. Correct? So c means what? We get the Km value directly where it cuts the y-axis. Sorry, x-axis. Correct? Graph gulo asche, don't worry, they are there. You have to draw a lot of graphs, don't worry. Significance of Km. I gave you 2 examples, glucokinase and hexokinase. You have any idea about these enzymes? Both the enzymes are the first and foremost enzymes of glycolytic pathway. Glucokinase enzyme is found only in liver, hexokinase is found in except liver and all tissue. Glucokinase is found in liver, Hexokinase is found in all tissues except liver. What is the difference between these two? Glucokinase is an isotype of hexokinase. It's type 4 isotype of hexokinase. What is the difference? They work on the same molecule, glucose to glucose 6-phosphate. One molecule of glucose attached with the phosphate of the ATP and forms glucose 6-phosphate, and ADP is released. Similar reaction. Site is different. What else? Their Km value. See the Km value for glucokinase and Km value for hexokinase. Which one is higher? Glucokinase. So higher means what? It requires more concentration of substrate to initiate the reaction. It contains more amount of substrate to initiate the reaction. Now, by name, what do you understand the difference between hexokinase and glucokinase? Hexokinase is relatively more broader specificity, whereas glucokinase is specific to glucose molecule. Now, the fallacy is, it is more specific to glucose, but it requires more glucose than required by the hexokinase itself to be triggered. গ্লুকোকাইনেজের কেএম ভ্যালু বেশি, যদিও গ্লুকোকাইনেজ কি করে মানে মোর গ্লুকোজ, জাস্ট বাংলা গুলিয়ে গেল। আচ্ছা, গ্লুকোকাইনেজকে অ্যাক্টিভেট করতে আরো বেশি পরিমাণ গ্লুকোজ লাগবে, যদিও এর স্পেসিফিসিটি গ্লুকোজের প্রতি বেশি, কিন্তু কেএম ভ্যালু বেশি মানে হচ্ছে তাকে বেশি পরিমাণ গ্লুকোজ দিয়ে তাকে অ্যাক্টিভেট করতে হবে। এই দুটো ভিন্ন ভিন্ন অর্গানে পাওয়া যায়। এর কারণ কি? এইটা হচ্ছে এই এত কিছু বলার উদ্দেশ্য। এবার আমার দেখতে হবে যে লিভারের কাজ কি। লিভার acts as a storage organ for excess of carbohydrate. When a carbohydrate will be stored in our body, when the carbohydrate is excess, so glucokinase is placed inside liver because when excess of carbohydrate is there, then only it will be stored. When carbohydrate is less, suppose my body requires 150 grams of carbohydrate, but I took only 50 grams of carbohydrate, so that will never trigger glucokinase to store glucose for future use. That will be utilized by our body. When it will reach the higher amount, say I'm taking 250 grams of carbohydrate, then that excess amount of glucose will trigger glucokinase to take up glucose and store for future use in the form of glycogen. If it was opposite, what would have happened? Even after taking food, we would have been hungry. Our satiety center would never be satisfied. Hexokinase যদি উল্টো দিকে থাকতো, তাই না? So Km signifies what? The amount of substrate required when it is towards affinity. Higher the Km, lower the affinity. Affinity মানে কত কম সাবস্ট্রেট কনসেন্ট্রেশনে সে কাজ করে। Km কি? তা ঠিক উল্টো, কত অ্যামাউন্ট অফ সাবস্ট্রেটে সে কাজ করে। তাহলে Km is inversely proportional to the affinity. Clear? এই ভ্যালুটা মনে রাখবে, 10 মিলিমোলার আর 0.2 মিলিমোলার। What is the unit of Km? Unit of substrate? কেজি, গ্রাম, মোলস, লিটার, যা খুশি, whatever will be the unit of substrate. Now we come to the last one, the types of inhibition. Okay, here a lot of things you have to remember. First, you have to remember the types of inhibition. Remember, based on the mechanism of action, there are two types: reversible and irreversible. Means which can be reverted back, which cannot be reverted back. Right? But how the enzymes, inhibitors, they react with the active site, based on that, it can be divided in this. We keep irreversible one separate, and reversible one can be of three types: competitive, non-competitive and uncompetitive. So reversible of 3 types and irreversible separate. It has no type deviation. It might have, but it's not so important because it is wasting the whole enzymes forever. Okay? So competitive, non-competitive, uncompetitive, and irreversible. So for each type, we will see the examples and the kinetics. For competitive inhibition, it is believed that inhibitor has structural similarity with that of the substrate. Which part of the substrate? The part of the substrate which binds with the active site of the enzyme. Right? That part of the substrate has structural analogy with that of the inhibitor. Right? So they compete for the same active site to bind. But if inhibitor binds to active site, it produces no reaction, no product. So formation of product will be less. Formation of product will be less. So rate of the reaction will be less. The reaction mechanism, you should take it down. The reaction mechanism is E plus S, ES, E plus P. Okay? Inhibitor binds irrespective of substrate. Correct? Irrespective of substrate. So it will bind to the same site where substrate binds. So it's like a musical chair. If you sit, your friend cannot sit. And if your friend sits, you cannot sit. So if inhibitor sits in the active site, substrate cannot. If substrate sits before, then inhibitor cannot. Right? But who will decide? Who will sit first? Concentration. Right? The apparent difference in the concentration. Right? If the concentration of inhibitor is same as substrate, then they have 50% chance. If it is 20%, then they have 1/5 chance. Okay? So this is E plus I, both arrow EI, because it is reversible. This is anyway, forget it. Draw this graph. Draw this graph. This is a Lineweaver-Burk plot. What is the difference in this graph and this graph? This graph, what is the difference? And with presence of inhibitor, it becomes more steep. Right? That means less reaction. That means when inhibitor binds, inhibitor is more efficient than the substrate. That's why it shows steeper graph. Now here the red line shows in presence the kinetics in presence of inhibitor. See? Sorry. Red line shows the normal one, and it is increasing. With blue line, see the increased concentration of inhibitor. Red line shows the normal one. This y-axis is a 1 by Vmax and x-axis is a 1 by Km. 1 by Vmax and 1 by Km. This point remains same. When you increase the concentration of inhibitor, what is changed? Vmax remains same. Km decreases or increases? Increases. Km increases, Vmax remains same in competitive inhibition. Correct? Example. Two such examples we will see. Two such examples. This is one of the very, very famous example of competitive inhibition. That is a well-known drug, sulfur drug, sulfonamide, is used to kill microorganisms. It is antimicrobial agent. Okay. So what they do, in all DNA synthesis, whether it's microbes or our human being, we require folic acid. We require folic acid. The folic acid is biosynthesized by the microorganisms, but humans cannot. We cannot, but they can. So in the biosynthetic pathway, this sulfonamide mimics one of the component of folic acid, PABA, para-amino benzoic acid. So during the synthesis of folic acid, this will occupy the space for PABA and folic acid will not be synthesized. In absence of folic acid or in deficiency of folic acid, the microorganisms or bacteria will succumb to death. And that's what we want. But will they affect us? No. Why? Because we do not synthesize folic acid. Right? So this is one of the example of using sulfur drugs which function as a competitive inhibitor in folic acid biosynthetic pathway, being structurally similar to that of the PABA, para-amino benzoic acid. Now the days have come, the winter days, to enjoy life at its full with high spirit. What do you mean by spirit? It means alcohol. Now alcohol consumption will increase. So poor people who cannot purchase good quality of alcohol, they often succumb to methanol poisoning. They often succumb to methanol poisoning. The methanol is very harmful for our body because it not only affects the liver, it also affects our optic nerve. People get blind. Now, in case of methanol poisoning, which you will find when you will work in emergency, particularly during the winter days in the rural area or suburbs, you will find patients coming with disorientation and intoxicated and having methanol poisoning, signs of methanol poisoning. Then what you will do? You have to keep pure ethanol in the ward, emergency ward. That is not for your consumption. It is not for your consumption. That is a therapy, kept for therapy. Okay. So you inject the same alcohol, or if he is in wake, you can ask him to drink, but it will be very drinking pure alcohol is very difficult. So you can inject alcohol and that will go and slow down the process of methanol metabolism, being structurally similar. It will function, it will inhibit the function of alcohol dehydrogenase. So now the alcohol dehydrogenase will not convert methanol to formaldehyde, which is more toxic, but rather it will convert ethanol to acetaldehyde. It will convert methanol to formaldehyde, but at lower concentration, at lower rate, so that our body gets enough time to get rid of that formaldehyde in the form of formic acid through urine. And our life will be saved. You understand what is the procedure? Right? So these are the two uses of competitive inhibition in your syllabus. You remember? Non-competitive inhibitor. The inhibitor does not interfere with substrate binding and vice versa. Unlike competitive inhibitor, it does not compete with the same sites. Means you and your friend has two chairs to sit. Correct? But if your two chairs are attached like a small bench, so now if your friend is as fat as me, then you cannot sit. He will occupy most of the bench. Similarly, in this case, they have two different sites, but within the same active site, but two different substrate binding sites for inhibitor. If the inhibitor comes, substrate has no point to bind. Even though they bind, the reaction will not take place. So what will be the kinetics? This is the kinetics. They bind, different binding place. Okay. Enzyme plus substrate, enzyme plus inhibitor, EI. ES plus I, ESI, EI plus S, ESI. from ESI does not produce product. Once it forms ESI, it does not produce product. Write down. This is all you have to write in the examination hall. This is the answer. This is the format, and then the kinetics, the graph. That's all. E plus S, ES plus E plus P, E plus I, ES plus I. In both the cases, it will form ESI. But from ESI, there will be no production of product. So the rate of reaction will decrease or increase. And what will happen to Km? It does not interfere with the binding of substrate. So Km non-competitive. Since it does not interfere with the binding of substrate, it has nothing to change in Km. Only thing will change is velocity of the reaction. See? Right? So velocity will be decreased and Km remains same. In competitive inhibition was Km altered, Km increased, and Vmax remains same. Here Km remains same, but Vmax decrease. Why? How do you remember V should decrease? What we are talking about? Inhibition. Inhibition means rate must decrease. Okay? So never V is never increased. Okay? Km might increase. That is inverse proportional. Allosteric inhibition. Example, you can remember allosteric inhibitors or allosteric. Actually, in case of allosterism, we do not say it is what? Inhibition or activation. We say modulation. Positive modulator or activator, negative modulator or inhibitor. So, example is AMP in fructose 1,6-bisphosphatase. This is uncompetitive. Okay. What are kinetics? The inhibitor binds only to the ES complex. Once the substrate is bound, then only it can bind. Right? Inhibitor can bind only after binding of substrate, not before that. This is called uncompetitive. How do you remember this? This is going to confuse you. Not only you, even few authors. In some books, it is written just opposite. What I am showing you, this is correct. But some books might have written it opposite. Knowingly or unknowingly, I don't bother. Okay? How to remember this? You remember, in this, inhibitor is binding to what? ES complex, correct? And in first stage, competitive, inhibitor was binding with free enzyme. So here it is binding with ES complex, and competitive inhibition, it is to bind with free enzyme. The previous one, the non-competitive, showed what? It can bind to anything. So can we call it mixed inhibition? So in some books, instead of writing non-competitive, they write mixed inhibition. So that this un and non terminology mistake or confusion never stays. So competitive, mixed, un. Competitive, un, mixed. Mixed means in both the side there will be EI formation. EI plus ESI. Both the sides. Inhibitor can bind with free enzyme, inhibitor can bind with bound enzyme. Yeah? Just see the graph and let's end. Hey, sorry, I'm sorry, extremely sorry. Since I have to somehow finish. The graph is like this, parallel. Both are changed. Km as well as Vmax.